Cloning is actually a scientific process known as somatic cell nuclear transfer. Somatic cell. Our bodies are made up of essentially two types of cells. There are germ cells, which are sperm and eggs, and somatic cells, which is everything else: skin cells, heart cells, liver cells, etc. In somatic cell nuclear transfer, what you do is you take the nucleus of that somatic cell and you transfer it into an egg cell that has had its own nucleus removed. The nucleus is where the genome is, all of the chromosomes that contain the A's and C's and G's and T's that make up the genetic code that make every organism look and act the way it does. Most of the time, when people do somatic cell nuclear transfer, they use skin cells. They have to go and actually physically take a piece of the living animal and grow the cells out from that piece that they use to make the clone. Our process is non-invasive. Instead of actually taking a piece of skin, we can take a blood draw, just a standard blood draw like an animal might get at a regular vet's appointment. And in that blood, there are cells. Unlike red blood cells, these endothelial progenitor cells have a nucleus with DNA, and unlike white blood cells, they don't carry multiple genomic rearrangements. And then we take the blood in that vial and we spin it down, and that separates all of the components of the blood by density. There's a fluffy layer on top, there's another layer on the bottom, and in the middle there's a little thin white layer called the buffy coat. This is where the cells that we are going to edit live. So we take that thin layer, we put it on a dish in some media that those cells really like, they start to grow up and differentiate, and now we have the cells that we're going to edit. Each cell is then injected into an egg cell where it develops into an embryo.